Okay, before we get started, I just want to give everyone a friendly reminder to not forget about the Connect questions that you have to answer for Chapter 11 and Chapter 12. I was looking in the Great Book on Canvas, excuse me, looking at the Great Book in Canvas, doesn't look like very many have completed both of those chapters. So if you haven't started working on it, you need to right away. Don't wait until next week, because those are two long chapters. There's a lot of mechanisms that are occurring at the molecular level in both chapter 11 as well as chapter 12. There are things you can't see, so be sure to read the reading assignments that are on the syllabus or in the Smart Book and do those questions. I'm afraid if you put it off until Wednesday or Thursday of next week, you won't have time to finish them, because there are quite a few questions, because these are long chapters. Okay, and I want you to all do well on the test, and that's the reason I'm having you do those questions, is that they'll kind of test your knowledge to see how well you understand the material. And if you miss a lot of questions, that should tell you, well, you need to spend more time studying. Okay? All right, so today we are going to start on the last section of chapter 11 that we're going to cover. This last section is on smooth muscle. Now, I would feel very negligent if I didn't talk at least a little bit about smooth muscle, because smooth muscle has some really different characteristics than skeletal muscle. And in fact, part of the overall mechanism of contraction of smooth muscle is different than skeletal muscle. The main part that's different is the control of contraction. In other words, the excitation contraction stage differs quite a bit from skeletal muscle. And again, in order to understand it, you have to look at what's going on at the molecular level. Okay, so there is some chemistry involved. All right, so smooth muscle forms layers within walls of hollow organs, and the smooth muscle that's in the walls of hollow organs undergoes wave-like contractions called peristaltic contractions that move substances through the hollow portion of tubular organs like the digestive tract. It's also found in the walls of blood vessels, and by contracting those smooth muscles in the walls of blood vessels, it could change the diameter of blood vessels, thereby changing the resistance to flow. And that plays an important role, as we'll see next semester, in the regulation of blood pressure. In addition, smooth muscles are also found in places like the iris of the eye. The iris is the colored part of your eye. Right in the middle of the iris is a hole called the pupil. And there's smooth muscles in the iris that control the diameter of the pupil. And that can control the amount of light that enters the eye. So if there's not very much light, it's really dark in a room, though some of the smooth muscles relax, the diameter of the pupil increases, that allows more light to come in. Also, there are smooth muscles in the skin. If you remember from we looked at the slides of the skin during about the third week of lab, we saw a type of smooth muscle called piloerector muscles. Piloerector muscles are little bands of smooth muscle that attach to hair follicles. And when your skin is exposed to cold, those muscles contract and it causes your hair to stand on end. Okay? So cold can stimulate some forms of smooth muscle, just like motor neurons can. And in fact, as we'll see, there are many, many different types of stimuli that can cause smooth muscle to contract. All right, so let's take a look at the structure of a typical smooth muscle fiber, or what are called myocytes. A myocyte simply means a muscle cell. It's a fancy way of saying muscle cell. So one difference between smooth muscle and skeletal muscle is in the number of nuclei per cell. Skeletal muscle fibers sometimes have hundreds of nuclei. Smooth muscle fibers, as well as cardiac muscle fibers, only have one nucleus per cell. And they can get by with the one nucleus because they're a lot smaller than skeletal muscle fibers. Skeletal muscle fibers are very, very long. They can be as much as 2 centimeters long. And so there's a lot of cytoplasm. And to be able to control all that cytoplasm, you need a lot of nuclei in skeletal muscles. Smooth muscle fibers are a lot smaller, they're a lot thinner than skeletal muscle, so they can get by with just one nucleus per cell, which is the typical, that's the typical number of nuclei in any cell. Usually there's only one. Another difference is that smooth muscle fibers lack sarcomeres. They do have thick and thin fibers or filaments, but the thick and thin filaments are not arranged in sarcomeres. They're arranged a little bit more randomly. And because of that, smooth muscles are not striated. And that's where they get the name smooth, is the fact that they don't have that striated appearance. So, another difference, a third difference, which has to do with the excitation-contraction coupling, is that in smooth muscle, the calcium that's needed to control contraction is not stored in sarcoplasmic reticulum. Smooth muscle doesn't have much sarcoplasmic reticulum. So instead, when a smooth muscle fiber is stimulated, there are calcium channels in the sarcolemma that open up, and that allows extracellular calcium to diffuse into the cytoplasm. So the source of the calcium that's needed to control contraction comes from the extracellular fluid and not from the sarcoplasmic reticulum. So that's one way that excitation-contraction coupling is different from skeletal muscle. And then another difference is that smooth muscle does not have Z discs. Since they don't have sarcomeres, they don't have Z discs either. Instead, the myofilaments, which are the thick and thin filaments, they're anchored to the sarcolemma, which is the cell membrane of the muscle fiber, by way of protein complexes called dense bodies. So that attaches the contractile proteins to the cell membrane. So instead of having the protein dystrophin linking the myofibrils to the sarcolemma like you see in skeletal muscles, you have dense bodies that attach the thick and thin filaments to the sarcolemma. The reason that smooth muscle fibers can still contract even though they don't have sarcomeres, which are connected to form myofibrils, is that the thick and thin filaments in smooth muscle are just, well, let me draw a picture here. Let me draw you a picture. In smooth muscle, rather than having sarcomeres, the thin filaments just overlap a single thick filament a little bit. So let's say that's a thin filament, then it'll overlap a thick filament, like right here. And then you might have another thin filament here, and then another thick filament. And then some of these thin filaments will be attached to the sarcolemma by way of a dense body. So that when the thick and thin filaments slide past each other and cause that whole arrangement to become shorter, that pulls on the sarcolemma and causes the whole cell to get shortened. Okay? So when smooth muscle is stimulated to contract, what happens is the thick filaments pull the thin filaments, like in that direction. And likewise at this end, it'll pull it this direction. And this one will pull, well, anyway, that causes that whole collection of myofilaments to get shorter. So you don't have a whole bundle of thick and thin filaments arranged in a sarcomere. Instead, you just have an overlapping arrangement of individual thick and thin filaments. Okay? And because of that, you don't get those light and dark bands that form the striations in skeletal muscle. Okay, now, as it turns out, there's two different functional types of smooth muscle. The first type, which is probably the least common, is called multi-unit smooth muscle. Multi-unit smooth muscle is found in structures that require a lot of fine control. And multi-unit smooth muscle fibers, or cells, are controlled by motor units. They have to be stimulated by a motor neuron. And so these are found in places like the iris of the eye, piloerector muscles, and just a few of the very largest blood vessels. And these are usually stimulated by a motor neuron that's part of the autonomic nervous system. Okay, it depends on where the blood vessel is located. If the blood vessel is located in a digestive organ, then it may be stimulated to contract by a parasympathetic fiber. But if it's in a different organ, sometimes they'll be stimulated by a sympathetic fiber. It just depends on the location of the blood vessel. So, an autonomic motor neuron has to be able to synapse with the fibers of multi-unit smooth muscle in order to stimulate that muscle to contract. Now the other main type of smooth muscle is called single unit smooth muscle. This type is most commonly found in the walls of hollow organs like the stomach and the small intestine and the large intestines. And it's called single unit because the individual cells in single unit smooth muscle are electrically coupled to each other. They're coupled by way of what are called gap junctions. Gap junctions are protein complexes that actually form a hole or a pore between two cells so that the cytoplasm of the two cells is actually connected. Since the cytoplasm of these cells is connected, action potentials can spread directly from one cell to another. So this is an example of what's called a chemical, this is what's called an electrical synapse. This is an example of an electrical synapse which you don't find in the nervous system. In the nervous system, the way that information is transmitted between neurons and effectors is by way of a chemical synapse. But these have electrical junctions. So when just one or a few smooth muscle fibers are stimulated to contract in single unit smooth muscle, the action potentials that those cells produce spread throughout that whole layer of smooth muscle, and it causes the whole layer to contract. So it acts as a whole unit. All right. Now, the contraction of smooth muscle, for the most part, is mainly involuntary. You don't really have control over it. And one of the reasons is that even the multi-unit smooth muscle is controlled by the autonomic nervous system. The autonomic nervous system is controlled by the brain automatically, and you ordinarily don't have any voluntary control over that. So you don't even have to be aware that it's happening, but your brain could be causing those smooth muscles to contract. But smooth muscle can also be stimulated by a variety of different stimulus types. It could be stimulated by way of the autonomic nervous system, by way of motor innervation, or it could be stimulated chemically by neurotransmitters or hormones like oxytocin. Some smooth muscle, like the fibers in the erector pili, can be stimulated by cold. Cold can cause the erector pili smooth muscle fibers to contract. There's quite a few types of smooth muscle that can also be stimulated to contract by stretch. There's some forms of smooth muscle, like there's some smooth muscle in the tubules of the kidney, that if blood pressure increases, which will cause there are blood vessels in the kidney that have smooth muscles. When blood pressure increases, that causes the diameter of those blood vessels to increase, which increases blood flow to the kidney. In order to regulate the rate of filtration, what happens is when those blood vessels stretch, that stretches the smooth muscle fibers, and that stretch stimulates them to contract, which causes the diameter of the blood vessel to decrease, thereby decreasing the flow of blood to the kidneys and regulating the flow of blood. So that's called a myogenic mechanism. It's due to contraction of smooth muscle in response to stretch. Also, there are some myocytes that can depolarize spontaneously. And they'll gradually depolarize until they get to the point where they reach the threshold, and that will cause an action potential to occur in that muscle fiber. That'll stimulate that muscle fiber to contract. So some muscle fibers can contract without being stimulated at all. So that's referred to as autorhythmicity. Okay? So there's a lot of different factors that can cause, most of which are autonomic or automatic. It occurs without you even necessarily being consciously aware of it. All right, now, the mechanism of contraction of smooth muscle fibers, in other words, the cross-bridge cycling, is essentially the same as it is in skeletal muscle. Okay? So what happens is that when calcium enters the cytoplasm of smooth muscle, that triggers a contraction, which involves a binding of the myosin heads to the thin filament, and then the myosin heads undergo a power stroke, and it pulls the thin filaments past the thick filaments, causing that whole arrangement of myofilaments to get shorter. Okay, so that cross-bridge cycling is the same. What's different, however, is the control of contraction. That is very, very different. So one difference is that smooth muscle lacks troponin. So calcium doesn't bind to troponin, and it doesn't have tropomyosin either. So when calcium enters the cytoplasm of smooth muscle fibers from the extracellular fluid, the calcium binds to a protein called calmodulin. Calmodulin is, so calcium combines with calmodulin, forming a calcium calmodulin complex. That calcium calmodulin complex then activates an enzyme that allows the myosin heads to break down ATP in order to release the energy from ATP so that the myosin head can be energized. Okay? So the controlled contraction of smooth muscle does not involve troponin or tropomyosin. Instead, it involves a couple of completely different proteins. Okay. Another difference is that the control of contraction in smooth muscle occurs on the thick filament, not on the thin filament. See, in skeletal muscle, the troponin and tropomyosin is on the thin filament. So in skeletal muscle, the control of the cross-bridge cycle occurs on the thin filament. In smooth muscle, the control of contraction occurs on the thick filament. Okay, now I think the best way to explain this is to show you sort of a flow diagram of the chemical reactions that activate a smooth muscle fiber to contract. So I think that I actually got this out of a different textbook, completely different textbook by a different publisher. So, in the first step, a stimulus, which may be a neurotransmitter released by a fiber of the autonomic nervous system, or it could be a hormone, or it could be cold, or could be any one of those stimuli that I mentioned earlier that can stimulate smooth muscle to contract. So the stimulus causes an action potential to occur on the sarcolemma, and that causes the opening of voltage-gated calcium channels, just like what happens in the synaptic knobs of motor neurons of the somatic division. So the stimulus causes voltage-gated calcium channels to open. Since calcium is in a higher concentration in the extracellular fluid than in the intracellular fluid, calcium then diffuses into the cytoplasm. Now, there is a small amount of sarcoplasmic reticulum in smooth muscle, but not very much. It's not very well developed. When the calcium enters the cytoplasm, that also stimulates calcium channels in the sarcoplasmic reticulum to open, and so that adds a little bit more calcium to the cytoplasm. But most of the cytoplasm comes from the extracellular fluid. So once calcium enters the cytoplasm, it combines with the protein calmodulin. This is calmodulin right here. So calmodulin is just always present in the cytoplasm of smooth muscle fibers. So calcium combines with calmodulin to form a calcium-calmodulin complex. Okay, it's just a combination of calcium and calmodulin. The calcium-calmodulin complex activates an enzyme that's in the smooth muscle fibers. This enzyme is activated by this calcium-calmodulin complex is called myosin light chain kinase. Now, a kinase is an enzyme that adds a phosphate to another molecule. So this enzyme that gets activated by the presence of calcium and calmodulin, it adds a phosphate to a part of the myosin protein. The part of the myosin protein is called the light chain. There's a light chain and heavy chain in myosin. So it adds a phosphate to part of the myosin protein, which forms the thick filaments. So this phosphorylated myosin light chain, then that enables it to break down ATP in order to release the energy in ATP. That energizes the myosin heads and allows the cross-bridge cycling to occur. Okay? So this calcium-calmodulin complex is sort of like flipping on a light switch that allows energy to be released. And that energy then is used to energize the myosin heads so that they can undergo the cross-bridge cycle. So if there's no calcium present in the cytoplasm, or very little calcium, then you don't have very much of this calcium-calmodulin complex, and so myosin light chain kinase is not activated. So without this calcium-calmodulin complex, myosin light chain kinase is turned off. It's in an inactive form. And without active myosin light chain kinase, then there's no ATPase activity of the myosin heads. So myosin can't break down ATP. If it doesn't break down ATP, then energy can't be released from that process to energize the myosin heads. So no cross-bridge cycling can occur if you don't have that calcium-calmodulin complex. Now, this may seem a little complicated, but it's really not, because there's really only 2 proteins involved. Well, 3 actually. There's calmodulin, which calcium binds to. So calmodulin is sort of analogous to troponin in skeletal muscle. Okay. Skeletal muscle calcium binds to troponin. That uncovers the binding sites for myosin heads, and cross-bridge cycling occurs automatically. Here, calcium binds to calmodulin. That activates an enzyme called myosin light chain kinase. That adds a phosphate to the light chain myosin, and that turns on the ATPase activity. So in smooth muscle, myosin can be turned on so that it can break down ATP and undergo cross-bridge cycling, or it can be turned off. If it's turned off, it can't break down ATP. If it can't break down ATP, then it doesn't have any energy, so it can't undergo a cross-bridge cycle. So this interaction here between calcium-calmodulin, myosin light chain kinase, that's sort of like an on-off switch that turns the myosin heads on, or if there's no calcium present, it turns it off. Okay, any questions? wait until somebody asks a question. I'm sure this is clear as mud, but one question you might have is, what are these chains that we're talking about? Well, this myosin light chain is actually a chain of amino acids that's part of the structure of myosin, okay? And there's actually two different polypeptide chains, or chains of amino acids that form myosin. And the one that has the myosin head is called the myosin light chain. There's another one called the heavy chain. So that myosin light chain has to be phosphorylated before the myosin heads can break down ATP. Okay. All right. Now, the phases of contraction of smooth muscle are also a little bit different than skeletal muscle. Remember that in skeletal muscle there was a short lag phase that is the time it takes for acetylcholine to go across the synaptic cleft. Also, it's the time it takes those ion channels at the motor end plate to open up, which allows sodium to go in. And then also, it's the time it takes for the calcium to be released from the sarcoplasmic reticulum. The latent period in smooth muscle, however, is about 25 to 50 times longer than that of skeletal muscle. So it takes anywhere from 50 to 100 milliseconds before a smooth muscle fiber can begin contracting. Then tension peaks after about a half a second, and then it declines over a period of 1 to 2 seconds. So everything about smooth muscle contraction is slow, basically. Lag phase takes 25 to 50 times longer. The contraction phase is slower, and it's also slower to relax. And it also has very slow myosin ATPase enzyme and slow calcium pumps that remove the calcium from the cytoplasm. That's why it takes so long for smooth muscle to relax. Now, smooth muscle also has some other unique properties. I like to say that smooth muscle is sort of a horse of a different color because it's so very, very different. It's like a purple horse. You don't see those very often. Smooth muscle also has what's called a latch bridge mechanism. And I don't think we need to get into the details of the chemistry of that right now. But basically, smooth muscle can contract and a latch bridge can form between the thick and thin filaments that can allow the smooth muscle to maintain its tension over very long periods of time without having to continue to use up energy in the form of ATP. So it can contract and it can stay contracted and it can maintain tension without having to expend a lot of energy. Another unique property is that smooth muscle can contract in response to stretch. So if you stretch certain forms of smooth muscle, that causes calcium channels to open. Calcium moves in, it binds to calmodulin. That complex activates the myosin light chain kinase, and that allows the myosin heads to break down ATP so that the myosin heads can be energized and they can undergo the crossfit cycle. So stretching can activate those calcium channels as well. And then another interesting property of smooth muscle occurs in the urinary bladder. The walls of the urinary bladder have has two layers of smooth muscle, and the urinary bladder can stretch a considerable amount, when it fills with urine. So if you didn't have a urinary bladder, you'd have to urinate all the time, just about continuously. Because we have a urinary bladder, we can store that urine until it's convenient to urinate, like in between classes. So when the urinary bladder starts to fill, those layers of smooth muscle in its walls stretch. And when they first stretch, they'll sort of fight back and try to contract a little bit, but eventually they relax. And then as it continues to fill, it can stretch more and more and more. And even when it's very stretched, though, when it comes time to activate the reflex, which is called the micturition reflex, that causes you to urinate, the smooth muscle can still contract, even though it's been stretched many times its resting length. Now, that's also very different than skeletal muscle, because skeletal muscle, if you stretch it too much, you pull the thin filaments away from the thick filaments, and you can't form any cross bridges any longer. Smooth muscle, since it doesn't have sarcomeres, it can be stretched a considerable amount, and as long as some of the thick and thin fibers still have at least some overlap, so the smooth muscle can still contract. So when the bladder is stretched, the tissue briefly contracts and then it relaxes, and then it stretches, it'll contract a little bit and relax. And eventually there's a micturition reflex that's activated, and the smooth muscle then will contract in order to empty the bladder. And that's also somewhat under voluntary control. The micturition reflex is controlled both voluntarily as well as involuntarily. Initiation of the micturition reflex occurs when you voluntarily relax the external urethral sphincter, which is made of skeletal muscles. That's voluntary, and you have to learn to control that when you're an infant in diapers. But once you voluntarily relax that external urethral sphincter, then an involuntary reflex occurs, which causes that smooth muscle to contract. So smooth muscle can still contract with considerable force, even if it isn't stretched at all, or if it's overstretched. And like I said, as long as you have some overlap between the thick and thin filaments, then it can still shorten. Another reason that it can stretch at just about any kind of initial length is that the thick filaments, the smooth muscles, have myosin heads along their entire length. So even if it's not stretched at all, you can still form a lot of cross bridges. Or you can stretch it quite a bit, and anywhere you have overlap between the thick and thin filaments, you can get cross bridges forming. All right, let's do just a few review questions here, and then we'll call it a day. This first question says, in which of the following organs, this is the code for this part of chapter 11. It's vismus, which stands for visceral muscle. So in which of the following organs would you likely find smooth muscle? By the way, next week, we should have plenty of time for review, so come to class prepared to ask questions, okay? Okay, let's take a look at everyone's response. Most of you said all the above, and that's correct, okay? Because remember, we said that smooth muscle is found in the walls of hollow organs. The stomach is one example of that. It's also found in the walls of blood vessels, controls the diameter of blood vessels. And it's found in the iris of the eye, as well as the ciliary body. Ciliary body is what allows your eye to focus by changing the shape of the lens. And then the lungs, we didn't mention that, but 3 of these correct, that must be all of the above, right? The smooth muscle fibers in the lungs are found in the walls of some of the airways, like the bronchioles. That can control the diameter of the bronchioles, therefore control airflow in different parts of the lung. Okay. Next question, which of the following is characteristic of smooth muscle? This should be a pretty easy question. Because there's only one correct answer on this one. Still have a couple of people that need to answer. Okay. Most of you said number 3, and that's correct. Smooth muscle can be stimulated by motor neurons of the autonomic nervous system. It can be stimulated by stretch, cold, hormones and other chemicals. And sometimes it doesn't have to be stimulated at all. Some smooth muscle fibers can automatically depolarize. What happened here? Okay. This one says visceral muscle is called smooth because of what? For what reason? Why is it called smooth muscle? As opposed to spindle-shaped muscle or something else. Let's take a look at your responses. Most of you said number 5, and that's correct. It's called smooth because it has no striations. Striations sort of make skeletal muscle look, has a rough appearance, and smooth muscle is basically uniform appearance. Okay, this question says, what part of smooth muscle contraction is most similar to skeletal muscle contraction? part that's the most similar is actually the cross-bridge cycling or the contraction phase. Okay, is that involves the binding of myosin heads to the thin filament, and then the myosin heads undergo a power stroke, and in order to be released, another molecule of ATP has to bind to the myosin heads. So I just kind of thought of an analogy. I don't know if this is a good one or not. Maybe you can tell me if it is. I just thought of an analogy for the difference between the control of contraction in skeletal muscle versus smooth muscle. Okay, so smooth muscle would be like having to turn on a switch to turn on the motor of an electric car. And that switch is in smooth muscle, is the activated myosin light chain kinase, which is activated by the calmodulin complex. Regulation of skeletal muscle, on the other hand, is more like putting up a barricade across the freeway that prevents you from going anywhere. That's what happens when tropomyosin covers up the binding sites. It's a barricade, and so cross-bridge cycles can occur. But in smooth muscle, the myosin heads basically can be turned on and turned off by myosin light chain kinase attaching a phosphate to that part of the myosin molecule. Okay, well, we're about out of time, so we'll stop here. And next week will be the third exam. Was that a good analogy?
Yeah.
I just thought of that off the top of my head.